Welcome back to DIY Biotech. Today I'm going to be giving a lab tour, and this is going to include a lot of DIYs or buys and sort of give you an introduction of how to set up a biotech lab. So first, let's get our PPE on. So you can see I've got these glasses side guards. I'll include a link in the description. These are really nice. They're way better than having big plastic safety glasses over your goggles. I also have nicely fitting gloves. They're not baggy. That's really important. I use these gloves, but every brand is going to have a different size that fits a little bit differently. Like this large fits me well, but there's other large gloves out there that are super baggy. So definitely check that out. And then I have a pretty generic lab coat on. So a big part of starting a lab is safety, and gloves are something that you use a lot of, so make sure you get those. A lot of working in a lab is just moving liquids around. So the way that I like to do that is with these Gilson pipettes. I'm not sponsored or anything. I would recommend getting a used set of these Gilson pipettes on eBay. They end up being pretty cheap, around like $50 maybe for some of them. And if you have pipettes, this is a P1000 pipette, so it goes from 100 to 1000 microliters. This is a P300 pipette, and this one happens to go from 10 to 30 or 20 to 30 microliters. This is a different brand. These both use very different tips, not only because they're different sizes, but because they're different brands. So I really like these Fisher brand Sure One tips to go with these Gilson pipettes. But these can be kind of expensive, but you can buy them pre-sterilized, which is really nice. If you need to work with larger volumes, you need one of these Serological pipetters. These are usually like rechargeable. You can probably find these used on eBay as well. And then you need to use these disposable Serological pipettes. These you can get for around 50 cents each on Amazon. I'll include links for everything in the description below. You can also get glass ones that are autoclavable. It really depends on your needs. But these can get pretty expensive. The 50 milliliter ones like this one are more expensive, while the 3 and 5 milliliter ones can be closer to like 10 cents each. Brought you in a little closer here to see one really nice piece of equipment to have. This is definitely on the nicer end of things. This is a shaking incubator. So you can have stationary things like Petri dishes up on this top rack, and then you can have liquid cultures in the bottom. So we have just about every possible piece you could have in a shaking incubator. We have these sticky mats, which I highly recommend. They're scary at first, but you can just put these shake flasks, which are baffled, so that they aerate properly, directly on the mats, and that's it. Whereas the metal ones, they're kind of a pain to pull out and put back in. Both the sticky mats and the shake flask holders are pretty expensive, honestly, but again, used on eBay as a potential. We also have an incline rack to hold these 2 milliliter tubes. And it's very important that you use culture tubes. So these are 14 milliliter culture tubes. It's very important to use culture tubes and not regular centrifuge tubes when growing cells. These are designed to improve aeration, and they have these special caps that are loose to allow air to come in. And then on the top up here, we have some Petri dishes growing. These are just some streak plates. And I'll include links to everything again in the description below. As far as DIY or buy goes, even a used one of these nice shaker incubators will run you a few $1,000s on eBay, and maybe tens of thousands of dollars brand new, depending on what model you're buying. So if you just need incubating space and not the shaking, you can use, I think the brand is Inkbird. It's just a temperature controller, and hook it up to a heating pad and then put it in like a styrofoam cooler. So you can build your own incubator for close to $50 or less, which is much more convenient than having the shaking incubator. If you need mixing, you could potentially use mason jars with a stir bar and a stir plate, and just use a small volume of liquid and put foil on top of the mason jar. And having that small volume of liquid and the stir bar will allow sufficient aeration of the culture. And you can just take that entire vessel and autoclave it just like you would a bioreactor. And we'll talk about autoclaving and sterilization in a minute. So these are two more really useful pieces of equipment. These are two scales or balances, whatever you want to call them. This one on the left does, up to a kilogram, so up to about 1,000 grams, and it's accurate to a tenth or a hundredth of a gram, so 10 milligrams roughly. But very often, especially for complicated media formulations or certain chemical reactions, you might need a more precise balance that goes to the milligram scale. Again, both of these I would recommend getting on eBay, but there's also some Amazon options that I'll include in the description if you need something just super cheap, like one of those jewelry scales can work well for you, but be careful of the accuracy of some of those. Here are another couple pieces of equipment that I think are really nice to have in a lab space that works with yeast or bacteria or any sort of biotech applications. This is a microcentrifuge. So this uses 1.5 milliliter tubes like these. So I'll include a link for these in the description below. But you need tubes that can handle the centrifugation. This one goes up to 16,000 G. So G is gravity, so 16,000 times gravity. It'll spin around and it holds 20 samples, I believe. So this, especially this Eppendorf brand, is pretty nice. I think we got this on eBay for like $200. They're not that expensive. If you're doing any DNA extractions, you need something that goes over 10,000 G. And then on the right here, this is a Vortexer. It's pretty fun. And this helps you resuspend cells. I don't think this is super necessary, but it's a very nice to have. And they're relatively cheap. I would recommend, again, getting a used one on eBay. That's really nice, but there's also affordable brand new ones on Amazon. In this lab, we also need to measure pH. I think that's a very common thing for biotech. This is an Amazon pH probe. This one's actually really nice, and I think it was less than $100. This over here on the right is a moisture content analyzer. We got this new on Amazon as well. It was a couple $100, I believe, and it works really well. It even talks to you, which is really cool. It's a very nice piece of equipment if you need to do any sort of dry weight analysis or moisture content analysis. So I'll go through a few of our consumables. I know this might feel kind of silly, but it is important to know all of these little pieces to sufficiently stock a biotech lab. So we have foil. I would recommend getting nice foil to cover any flasks or cover any ports on a bioreactor if you need to autoclave that. We have weigh boats, plastic weigh boats, and metal weigh boats. So if you need to dry something, you need to use the metal weigh boats. Stir bars. We have Ziploc bags. We have a laser thermometer gun, a toolkit, and hearing protection. So we have a ultrasonicator, and it's good to have that hearing protection. We also have like a grinder, a spice grinder to grind up biomass for the stuff that we do. So it's good to have hearing protection. And then a toolkit is always essential for a lab, very underrated. We are fortunate enough to have this huge, really nice, chilled centrifuge. This can hold up to 250 ml bottles, you can see there. But it also has baskets. These are removable baskets that can hold 50 milliliter tubes, and you can also buy ones that hold 15 milliliter tubes. So these are some 50 milliliter tubes. Very good to have these depending on what you're doing, but 15 milliliter tubes are also useful as well. These centrifuges are pretty expensive, so unless you really need to do larger scale stuff, I wouldn't recommend getting one of these. But typically these go up to 4,500 G, that's the max setting on this one, and they can process liters at a time. This one also has a cooling function, so it can get down to about 2 degrees Celsius, which can be useful for protein extraction. Some other small pieces of equipment you may need. This is a microwave, also a very underrated piece of equipment. We have a vacuum chamber up there for drying samples. And then we have a hot water bath for keeping things at a constant temperature. You can also get something like a heat block, and a heat block might be better if you're doing smaller volumes that need to change temperature quickly. Here is another very nice piece of equipment to have. If you're measuring cell growth of any kind, or even protein expression, or DNA or RNA, you need some way to measure those samples quickly. So this is a cuvette holder. So you can use one of these cuvettes like this, and this holds down to 500 microliters of sample at a time. And it just goes in like that, and then it slides in and it shines light through. And it has variable wavelength, so you can measure the optical density of cells, which is a proxy for cell growth. You can measure protein, you can measure DNA, you can measure RNA. But if you need to measure DNA or RNA, if you're doing any genetic engineering, you need to have a nanodrop. And I would recommend getting those used on eBay, but that's going to be in the $1,000 price range. And you can literally use a single microliter, like smaller than a drop. It looks smaller than a drop, and it will tell you how much DNA or RNA or protein is in a sample. So very useful piece of equipment. This one also has a plate reader. There's tons of different plate readers and cuvette readers and nanodrops out there. I don't necessarily recommend this one, but it works fine. As far as chemicals goes, this is going to depend a lot on exactly what you're doing. If you're working with yeast and bacteria, your chemicals may look pretty similar to mine here. So if you're working with yeast, you probably use YPD, which is a media that contains yeast extract, Peptone, and Dextrose, aka glucose. You also are probably making petri dishes, so you need agar. These are pretty standard chemicals. I would recommend getting nice brands of these chemicals. You want consistency batch to batch, and you want agar that's actually going to solidify properly. For any liquids that you have, it's nice to have secondary containment in case something leaks. These Fisher brand 1 liter bottles are super, super nice. They're autoclave safe, and they're really not that expensive. We have 1 liter bottles, 250 and 100 ml bottles, but it depends on your needs. We also have some other chemicals up here for particularly what we're doing. We have like xylose and ammonium sulfate as a nitrogen source. These yeast nitrogen bases are really expensive, but they're very nice and reliable. This is sodium acetate, I believe, yeah. So I'll go through some of these commonly used supplies. These are cryovials. So if you're making any glycerol stocks for mammalian cells even, but yeast cells, bacteria cells, maybe even insect cells you're working with, you mix glycerol with your cell culture, and you can freeze them in these cryogenic vials at -80 Celsius, which we'll look at in a second, and that will preserve them. They'll preserve the cells nearly indefinitely. It's nice to have like a box cutter. These particular Sharpies are indelible, so they'll write on the plastic tubes and such, and not really come off, even with ethanol, which is very, very nice to have. But don't use these to write on paper or they'll wear out quickly. I really like this Fisher brand tape too. It survives autoclaves, but it comes off bottles nicely. I have a replica plater here. This is a nice to have. Basically the way that this works is you get one of these velvets and you put it on top, it's sterile, and then you put this ring around it, and then you take your plate, your agar petri dish, put it on top, and you get a different petri dish and you put it on top, and it sort of copies the plates. It's a replica plater, it replicates the plates. Kind of expensive, but pretty cool to have. Let's see what else is in here. More cryo vials, some 15 ml tubes. Brand doesn't really matter for these 15 and 50 ml tubes. Parafilm, that's a nice to have. You don't need to have that. Sterile cotton swabs, which we're currently out of. Let's see what else. We have a bunch of these serological pipette tips and these culture tubes. It's very nice to have these sterile culture tubes, but they do get kind of expensive. Very, very, very important for a lot of microbial work. So we have a couple of cold storage units here. This is just a 4°C fridge. So you could just find that on the side of the road or on Craigslist for $20 if you need just 4°C. We actually don't have a -4°C or a -20°C freezer, so everything just goes straight to -80. So yes, this is -80. It's about as cold as liquid nitrogen, and the samples freeze very solid and stay good forever. These are our glycerol stocks in those cryo vials that I was telling you about. So they stay good about indefinitely. So that's our cold storage there. Even small versions of these -70, -80 degree freezers are pretty expensive, in the few $1,000 range. Maybe you can get one for $500, but they are very, very useful. So these are two of my favorite pieces of equipment, honestly. They're really nice, but very not necessary for a lot of different applications. This one is a rotovap, so it uses this glass vessel and a hot water bath, and it pulls vacuum on this column to gently evaporate solvents, including water. So that can be very good for production of delicate chemicals like carotenoids or something like that, that you don't want to boil the liquid away. Similarly, if you want to keep your sample pretty stable without, and you know, get rid of some solvent like water without damaging your sample, a lyophilizer is really nice. This is also a very expensive piece of equipment, but there are cheaper ones out there, and there are some DIY options out on the internet that you can do. But basically, this has a chiller inside of it that goes to -80°C, and then it pulls vacuum as well to freeze dry the sample. So it's a lyophilizer or a freeze dryer. It's very useful for producing pure proteins. So without degrading the proteins, you can actually freeze the protein sample, put it in the freeze dryer, and then all of the water sublimates off, and you have a pure protein sample. So very useful if you're doing protein sort of stuff, or protein purification, that could be useful. Or if you need like cells at the end, like clean cells at the end of a process, that can be useful. And this can be useful for delicate samples as well. But this is probably $1,000-ish even used, but this is a really nice one. And then this is a few $1,000s as well. I know I said those two were my favorite pieces of equipment, but this is my child right here. This is my baby. This is our 2-liter bioreactor. Really cool, really shiny. This is an Eppendorf bioreactor. We got this used on eBay for about $15,000, and it came with a chiller as well, which is down here. So we got a really good deal on those, and you can lowball people on eBay and get some pretty nice equipment. But the way that this works is it has a controller. So this controls pumps that go to the bioreactor. It controls all the sensors. It records all of the data. It maintains the temperature, makes sure the pH is good. This bioreactor is, I do not have all of the probes with me right now, but there's a pH probe, a dissolved oxygen probe, there's a sample port where you can take samples out, there's a temperature probe port. Pretty standard, pretty easy to work with bioreactor. So this is an Eppendorf bioreactor. I like it. It also has this motor that has really fine speed control that mounts on the top, that turns an impeller inside. It's got this heating blanket to control temperature. And if you like bioreactor stuff, I will be doing a video in the next month or so on a DIY or buy for bioreactors. So this is sort of mid-buy level, so used, $15,000. If you want brand new ones of this size, they can go up to like $50,000. If you DIY, you could potentially make one for $50, somewhere around there, depending on what you consider a bioreactor. So subscribe to the channel if you want to see more like that. So finally, let's talk about sterility. This is a proper autoclave. It's huge. It's massive. I'm 6'4". This thing is huge. I could fit in there. So this is really great if you need to autoclave up to even like a 20-liter bioreactor. It could fit in this autoclave. But these are probably $100,000, so not very DIY of me. But this is as big as you can go. This is basically just a giant instant pot or a giant pressure cooker. So if you want to autoclave things, as long as they're less than like 500 milliliters, you can just buy a big instant pot, and I'll put a link for that in the description below as well. I use that at home all the time. It works fine. You just maybe need to pressure cook it for a little bit extra time. But to keep things sterile, you have a few options, and I'll give you some levels here. So the bottom level is just an ethanol flame, or you can even really use a candle, which I've used in previous videos. And it is just a little flame, And around it, you have this sterile field because the heat from the flame pulls air up to prevent contamination from falling down. This does lead to more contamination than using an appropriate piece of equipment, but it's good enough for yeast and bacterial work. If you're doing any fungal work or plant work, you at the very least need to have a laminar flow hood. So the way that this works, we'll go over a few pieces of equipment, but the way that the laminar flow hood works is it blows sterile air over your sample. So it draws in air from the top, it goes through a sterile filter and over your sample. The way that you work in this is you want your most sterile stuff to be closest to the airflow and your least sterile stuff to be in the front. This takes skill to work with. You need to make sure not to pass your hand over open bottles and things like that. But this is going to keep things a bit more sterile than an open flame. So that open flame, you can get one of those for 20 bucks or less. Again, I'll include links in the description below. Something like this, you can DIY one of these for $100 or $200 with something like a box fan and a HEPA filter. Or you can buy a proper one for probably on the order of $1,000. The next piece of equipment is back where we started. This is a laminar flow hood. This keeps you safe to some degree and your sample safe. If you're working with mammalian cells or insect cells, you need to have one of these laminar flow hoods. You will not be able to keep your work sterile without one of these, and these are a few $1,000s. If you're working with yeast and bacteria, this is definitely overkill, but your samples will almost never get contaminated if you use just simple, common, sterile technique. This is really nice. You have to set the sash to the exact height that is prescribed by the machine. You give it a few minutes, and it blows sterile air in a laminar way. Some of the air comes out, and some of the air comes in. I'm not a designer of these, but basically it's a biosafety cabinet, so it protects you and the sample to some degree. Very, very good. I've almost never gotten contamination in one of these. It's also very nice to have these ethanol spray bottles for sterility. So in here we have 70% ethanol, and anytime we want things to be sterile, we just do a couple of squirts, rub it on our gloved hands, or rub it on our pipettes, or rub it on our tubes, and it will basically make things sterile. You can spray your whole work surface down. You can spray any tools that you're going to be working with. You can spray that down in ethanol. But again, the degree that you keep things sterile is really going to depend on what type of cells that you're working with. This lab is mostly applicable to yeast and bacteria. A couple more small things. This is a microscope, really nice to have. It goes to 1000x. So it has these 10x eyepieces, and then it has a lens that goes down to 100x, so 100 times 10 is 1000x. It also does 40x at the smallest magnification. Pretty cheap, like 150 bucks on Amazon. I'll include a link in description. And then you need these microscope slides and these cover slips, which are also pretty cheap, just like $10, $15, something like that. If you want to use the 1000x objective, you might want to use some microscope immersion oil. That might be good to have. And these chem wipes are also nice. These are basically just lint-free wipes. They're pretty expensive, and I almost never use them, but it depends on what you actually work with. So here is the scary, dangerous stuff. This is a proper lab space, so we have acid cabinets and flammable cabinets and strong base cabinets. I would recommend keeping those three things separate no matter what lab space you're working in, and keep them in secondary containment. Up here we have our chemical fume hoods. So we've looked at laminar flow hoods, which keep your samples safe, biosafety cabinets, which keep you and the sample safe, and then chemical fume hoods, which just keep you safe. So it pulls air in and up and away. So this is where you want to work with solvents, anything flammable or strong acids or anything like that. You work in these biosafety cabinets. A DIY version of this might be an open window and a box fan, but I'll leave the safety up to you. We have very serious safety restrictions in this lab space, so we have to use these pieces of equipment properly. So I'll end on this piece of equipment here. You almost certainly do not need one of these. It's a microwave reactor. It's a few $1,000s, even used. But I just want to emphasize, your lab build out may look very different than mine, but I think a lot of the pieces that I showed today are pretty fundamental, especially like the pipettes and the consumables like the tubes, and just general lab safety and sterility. But if you're working with yeast and bacteria, I think your lab will look a lot like mine. Once you start working with DNA extractions or any genetic engineering, you might need specific DNA extraction kits. Or if you're working with mammalian cells, you might work with different consumables. But that's going to be something that you can discover along the way. I think this video is a really, really good starting point. If you want to see things like this or have any questions or comments, leave them in the comment section below. And again, just about every piece of equipment or consumable that you see in this video is going to be linked in the description below. They are Amazon affiliate links. It helps the channel out a lot, especially if you want to support the channel. Go through those links and do your lab shopping. That would be super helpful. So again, don't forget to like the video and subscribe to the channel. Thank you so much for watching. Bye.